Hi, I'm Erik Feldmann. I'm a technical director at Cambrex. I'm located in Durham, North Carolina, and I'm responsible for the early stage business unit. So for peptide sequencing, the classical way that the companies have used peptide sequencing would be Edman degradation. Edman degradation is inefficient, and there have been a lot of advances in mass spec technology, in particular LC-MS with tandem mass specs, that's MSMS. And the reason for utilizing peptide sequencing in pharma would be to ID a particular species of polypeptide. So in the biologic space, this may be a small peptide, this may be a large polypeptide like a protein, and if it's a protein, there could be different types of proteins like an enzyme or a monoclonal antibody or a polyclonal antibody. Peptide sequencing itself can take different approaches based on how big that polypeptide sequence is. If it's a protein, in this case if it's an antibody, they could be really large, and they may be too large for the mass spec instrumentation that you're using. And so it requires extra steps. In this case, you may need to have a protease digestion to separate out all of the peptide sequences, and then you have to have a chromatographic step to separate all of those, and then the mass spec detector that can ID each of those peptide fragments, okay? Together, when you've sequenced all of those fragments together, you then map the coverage of all of those peptide sequences into that full polypeptide. That would be the ID of your material, and that's how peptide sequencing is being used. So I think the technologies right now that are revolutionizing peptide sequencing in the field would be advances in the software and the data mining. So the mass spec technology that Agilent and Thermo Fisher and Waters are developing utilizes library matching and searching of known peptide masses. And so with these libraries, you can very quickly identify what the sequence is when you're looking at the data output. So that in itself, the separation of the peptide sequences is step one. Collecting of the mass spec information is step two, and then library matching is step three, and that is automated. So that makes it much more efficient because you can inject multiple samples, separate all of those on your high-throughput HPLC system, collect all of that data with a high-res mass spec detection instrument, and then the library itself will match all of those data outputs very quickly. So for efficiency, right now that is the emerging technology that gives you that high throughput ability to manage multiple sequences, multiple samples in a given data set.